Go ahead.
Thank you, and thanks to the organizers, and thank you, Evelyn, for inviting me today to present my work at this nice virtual meeting. So I'll be talking about numerical methods that can be employed to perform 3D detailed simulations of neuronal processes. The work, I'd like to mention the collaborations that are involved in some of the work I'll be talking about today. I'd like to point out the collaboration with Andreas Vlachos. In this context, most of the work I'll be showing today is in collaboration with Marcus Bright and Martin Stepniewski, as well as James Rosado at Temple University. So the intracellular scale that we're interested in here is upon electrical activities of individual neurons. As you probably know, synaptic regions are activated, inducing calcium entry at synaptic sites. And there's a big question of how calcium signals then can propagate from remote regions in the dendrites down to the cell nucleus, which is harbored in the soma, where calcium signals then activate transcription responses at the nucleus and endoplasmic reticulum that are relevant for learning, plasticity, survival, etc. So at that scale, the three-dimensional architecture becomes relevant, since there are organelles like the endoplasmic reticulum and mitochondria that function as large calcium stores and exchange calcium across the organelle or plasma membrane. And these dynamics then induce calcium signals that can propagate over longer distances. So this is in general what we are interested in studying. And looking at the ultrastructure of individual synaptic spines, here are a couple of examples that were reconstructed from raw data from Mark Ellisman's lab and in this project. And as you can see, the architecture of individual synaptic spines is fairly intricate, with actin filaments filling the space, with organelles like the endoplasmic reticulum being embedded in the spine heads. As we see here, the postsynaptic densities at different regions. All of this architecture is involved in communicating calcium signals from the spine head to the dendrite and then on towards the nucleus. So we'd like to know how these complex and varying cellular structures influence the calcium dynamics towards the nucleus. So the first thing one has to consider is a model for intracellular calcium, which is schematically depicted up here. And from there, using a model, which I'll point out the details in a second, we want to use detailed three-dimensional architecture to study the influence of structure on calcium function. So over here, we see an example of electron microscopy data that is used to then identify the individual components of, let's say, a spine consisting of spine head, neck, piece of the dendrite. In green, the endoplasmic reticulum or spine apparatus, which is then connected to a part of the dendrite in this case. And as you can see on the right, morphology varies. So there are different types of spine morphologies. Some are termed stubby spines, others are termed mushroom spines. And many of them contain a piece of the endoplasmic reticulum that innervates the spine head. Now, having techniques to reconstruct the three-dimensional architecture of these components of a cell and the organelles, we can couple this to a three-dimensional model of calcium dynamics. Now, the first ingredient here is the fact that calcium can interact with mobile buffers in the cytosol, which is this yellowish region. We assume that calcium in the cytosol can propagate via diffusion and reacts with mobile buffers. So we also track the mobile buffer concentration B over time, as well as the cytosolic calcium concentration. And this can be then combined with the enzymatic production of a molecule called IP3, which then can interact with so-called IP3 receptors. So these are the components we track in the cytosol. And the green region is the endoplasmic reticulum. And inside the endoplasmic reticulum, we assume that endoplasmic calcium can diffuse. So there is work that shows that the endoplasmic reticulum seems to be a connected organelle, so calcium can freely move within that organelle. So we assume diffusion. Now, the boundary conditions are basically what drive this calcium system. So on the one hand, we have exchange mechanisms on the endoplasmic membrane. These are ryanodine receptors, IP3 receptors that pump calcium from the ER into the cytosol. And on the other hand, there are circuit pumps which replenish the ER with calcium that's available in the cytosol. On the plasma membrane, there are similar pumps and exchangers like PMCAs and sodium calcium exchangers, voltage-dependent calcium channels that operate at the plasma membrane. So all of these exchange mechanisms, which then are formulated as nonlinear boundary conditions to this PDE model, can be incorporated to simulate the propagation of calcium signals driven by these exchange mechanisms. So I'll just briefly highlight the different types of exchangers like IP3 receptors. I won't go into detail. This is published work that now is integrated into a three-dimensional model. So IP3 receptors, circuit pumps, ryanodine receptors are all models for which one computes the flux across the boundary, which can depend on the endoplasmic and the ER and the cytosolic calcium concentrations, making these nonlinear terms. On the plasma membrane, similarly, I mentioned a number of pumps that take calcium from the cytosol and pump it into the extracellular space. And on the right-hand side is a table that I forgot to mention the reference here with the parameter set that we use for a specific study of calcium dynamics in spines. Now, all of this requires solving PDE-based models. So in the previous talk, there was a focus on stochastic differential equations. Here we're dealing with deterministic models, so everything is described by partial differential equations. And if we remember the complex structure of spines or the shape of neurons, discretizing these PDEs, making them finite dimensional, on a discrete computational grids typically leads to very large linear systems, since we have to represent this complex geometry, which leads to very large linear systems that need to be solved. And the size of these linear systems are a point of interest, since direct numerical solvers for this problem size are not feasible. And iterative methods, for instance, Jacobi or Gauss-Seidel iterations, tend to be too slow for the system size. So one typically turns to hierarchical methods, which are iterative methods, but they exploit a grid hierarchy, which, if derived and implemented correctly, produce H-independent convergence, H being basically the smallest grid size that one encounters in the problem. So this would be optimal convergence, which is ideal for the system size we're interested in. So one representative of hierarchical numerical solvers are geometric multigrid methods. So on the right, you see a small schematic of a two-grid idea. So I'll just quickly go over the idea of geometric multigrid, and that is we're looking at a problem on a fine computational grid, omega sub h, but we don't intend to directly solve the problem on the fine grid, but instead exploit a coarser grid. So we move from a fine grid to a coarse grid and try to compute a correction to our solution or approximate solution on the coarse grid, which is much cheaper computationally spoken because the number of degrees of freedom on the coarse grid is far reduced. And of course, one could build a whole hierarchy of more than two grids to go down to the coarseness required to directly solve a problem on the coarse grid and use it as a correction on the fine grid. So given a linear problem, which we'll call LH times UH equals FH right-hand side, we're interested in solving for U sub H. And geometric multigrid, or in this case, two-grid method, one begins with a pre-smoothing step. That is, you take the current iterate of your solution, you apply a smoother, which typically is one of the iterative solvers, which we don't use as a solver, but we just exploit it as a smoother, which gets rid of high-frequency error components on the fine grid. So this is cheap because we only apply 2, 3, 4 smoothing steps before we then compute the defect, which is defined in this way on the fine grid, but then restrict the defect to a coarser grid. So this restriction operator here merely restricts the fine grid defect to the coarse grid. Then we solve the coarse grid correction. So we solve this equation, which should be cheap because we're on a coarse enough grid. Once we've done that, we can prolongate the coarse grid correction back to the fine grid by some prolongation operator. All these ingredients have to be defined and chosen wisely. And then we update our current iterate with the coarse grid correction, producing the solution U tilde, to which we apply another post-smoothing step on the fine grid. So the geometric multigrid approach consists of a smoothing operation on the fine grid and a coarse grid correction. So the caveat to this optimal convergence that I mentioned is that grids need to be regular enough. That means grid quality matters here. And oftentimes when dealing with microscopy data and reconstruction techniques from microscopy data, one encounters ill-shaped elements in the computational domain. So element angles and domain interfaces influence the numerics here and make it less than optimal. And once we've introduced by reconstruction method, for instance, elements that are unfavorable for the numerical approach, these would, in a multi-grid method, when refining, just propagate through the grid hierarchy. So the question is, how do we automatically deal with effects that can happen due to grid quality? And one approach we took was to use so-called smooth subdivision theory to optimize volumetric computational grids automatically that work with geometric multigrid methods. So here's a visual idea of subdivision methods. So one starts with the coarse grid and continuously applies an operator S to the grid, which then produces or converges to a limit surface in this case, and this can be done for volumetric grids as well. So given a coarse control polygon, we repeatedly refine the grid, but then we reposition vertices with distinct position masks, and these are defined by this operator S, which is called the subdivision matrix, which encodes the vertex insertion and repositioning in each iteration. While there's a lot of work for subdivision surfaces originating in computer graphics, there's much less theory when talking about volumetric grids. So we had looked at this more closely since we're interested in optimizing volumetric grids. And this can be also done if one chooses a wise element splitting, for instance, from tetrahedra to a mixed grid consisting of tetrahedra and octahedra. So in that case, one can show convergence for this method, and we then implemented this subdivision volume technique, which is illustrated here, which consists of insertion of new vertices, repositioning based on the element type, and creating in that way a hierarchy of grids, which then in a final step, one can project back to the coarsest level to ensure node nestedness, which for many of the available codes is one requirement that one has nested grid hierarchies. So here are two examples where we chose a unit cube and a cell nucleus, in which we then artificially decrease the quality of the grid by moving this vertex further to the boundary in both cases. And what we see is that with decreasing grid quality in panel B, we see, of course, an increase in the number of iterations needed for convergence. So with decreasing grid quality, we observe what we'd expect, a decrease in the efficiency of the numerical solver. Now, this is the case for standard geometric multigrid in blue, whereas if we apply the subdivision geometric multigrid I just highlighted, we can see that the number of iterations can be held in check by automatically regularizing the computational grid. And in these cases, and there are some cases, as you can see, for the cell nucleus where the standard method diverges and the subdivision geometric multigrid method performs very well, and we can, in some of our test cases, see improvement factors of over 50 when comparing this SGMG method with standard geometric multigrid. So having this regularization technique in place, we can deal with complex geometries. So I'd like to give a brief example of calcium dynamics and a study we performed using these techniques. So we're interested again in a spine region that's connected to a piece of a dendrite. Embedded is the endoplasmic reticulum that can grow into the spine itself. And we looked at what is the role of the endoplasmic reticulum in synaptic spines. So we started out by designing a geometry which only contains dendritic ER and no ER in the spine. And then we grew the ER further and further into the spine. And first of all, just considering the ER to be passive, so no exchange mechanisms are active on the ER membrane, we see that the ER mostly functions as a small diffusion barrier. So you see slight variations in the calcium amplitude depending on the choice of ER. So with increasing ER in the spine, we see some diffusion limitations, but it's very minor. Now, if we activate ryanodine receptors and IP3 receptors and we repeat the same thing, we can see that under no ER conditions, we do not observe any dendritic calcium response when providing a single pulse at the spine head. So we see that in the head and the neck, we see a calcium response, whereas at the dendrite, there is only a response, excuse me, I was pointing in the wrong direction. This is the neck, this is the head. And in the dendrite, we only see a response if the length of the ER into the spine head is sufficient to induce spine to dendrite coupling. Now for IP3 only responses, we see that IP3 receptors increase cytosolic calcium at a much longer time scale and not to the extent as ryanodine receptors do. And for IP3 only, we don't see a response in the ER. Then coupling both, we see that again, given specific ER configurations, we see a dendritic response, and adding IP3 receptors, one can observe protracted responses in the dendrite due to this slower elevation of cytosolic calcium through IP3 receptors. So ryanodine receptor calcium trigger communication, length, spine to dendrite communication. We see signals with IP3 receptors, and the length also influences how less and one can reverberation in the spine hair, in the spine neck, rather combining the two receptor types. Now, finally, excuse me?
The last bit was not understandable. Your connection seems to be rather, at least it wasn't very good at the moment.
Okay, is it better now?
Yes.
Okay, so the last part I was mentioning is that taking ryanodine receptors and IP3 receptors together, we see the previously observed coupling if the spine, if the ER length is sufficiently long, but we also see reverberations in the dendrite and the neck if one adds IP3 receptors, which was not the case for ryanodine receptor only signals. Now, a last thing I wanted to point out is the question about spine head growth. So over time, spines adapt their shape and grow. And what happens under spine head growth to calcium signals? In particular, can a reorganization of the endoplasmic reticulum ensure calcium homeostasis in the spine head? So we start out with the reference spine and observe the calcium signal in the head, the neck, and the dendrite here. But increasing the spine head volume by a factor of 2 then disrupts the connection between the spine head and the dendrite. So we do not see any dendritic calcium response anymore under spine head growth. So I thought, well, can we grow the spine, can we grow the endoplasmic reticulum further into the spine head to induce coupling? And in fact, yes, with that, we see that we can recouple the spine head and the dendrite, although we don't get back the original calcium profile. It's similar, but not identical. So we then increase the volumetric part at the tip of the ER. And with that modification, one can, comparing B and E, see that under spine head growth and reorganization of the ER, one can guarantee calcium homeostasis in all these three units, head, neck, and dendrite. So upon head growth, calcium concentrations drop, as one would expect. And we see that ER repositioning can restore calcium, the original calcium signal, to a certain extent. And therefore, calcium homeostasis can be regulated by intracellular ER reorganization. And upon completing this study, we were looking at the fact that we had to increase the volumetric part of the ER in the spine head to ensure this calcium homeostasis. And what could be observed in published work is that in fact, once the ER grows into this head, to grow in such a way to be the surface to spine head. So these geometries seem to replicate the prediction of the model here, that one to do exactly what is observed in order to ensure calcium homeostasis under geometric changes of the spine itself. So we are currently working on reconstructing human dendritic spines and analyzing the effect of actually reconstructed spine morphologies on calcium dynamics. So what we saw in this model study using idealized geometries, does that transfer to actual spine reconstructions? And here's an example of a simulation that shows that under calcium release in the spine head, calcium-induced calcium release through the ER can actually induce calcium spiking in the spine neck, independent of new input from the spine. And this is all purely morphology driven, as other spines do not show this type of behavior. So these last spikes you see in the neck are auto-induced by these calcium-induced calcium release mechanisms that I showed at the very beginning. Okay, so with that, I highlighted the fact that fast numerical solvers are necessary to solve these large linear systems that arise from PDE-based models in 3D, exploiting detailed three-dimensional morphology. I presented ways to automatically regularize geometries or computational grids to include them efficiently in hierarchical numerical solvers like geometric multigrid, and then incorporate detailed reconstructed morphologies into numerical simulation to study the influence of 3D structure on, in this case, calcium function. And with that, I'd like to close and thank everyone for joining. And again, thanks to the organizers for organizing this great meeting. Thank you.
Thank you, Gillian, for this interesting talk. We have a question by Julia Timofeeva. I expect the modeling results on ER will be affected by the choice of densities of the RyR and IP3 on ER. Are the experimental data data available to constrain these densities?
There are some experimental data that indicate whether or not a specific receptor type is present at the spine apparatus and the endoplasmic reticulum. And there are some ballpark figures, some ballpark numbers that can be derived from estimates of the surface area and numbers that have been observed in different systems. Although I have to say that really reliable numbers are not, or to my knowledge, not available so far. So in all of the studies, we've used these ballpark computations and varied the parameter space significantly to determine the influence of the choice of, let's say, a ryanity receptor density or IP3 receptor density on the outcome of the simulation. Yeah.
Okay, thank you. I'll switch you off